Good morning, students. Now we are going to discuss into an important concept is structure of Anther and structure of microsporangium. In PUC examination, it is one of the important 5 marks question. In our NCERT textbook, for the structure of microsporangium, only given to 5 to 6 lines only. But here most important points are involved in the structure of Anther. Generally, before class already we are discussing about the Anther. You know that the Anther, in that Anther, 4 wall layers are present. How to that 4 wall layers are developed in that Anther? First of all, initially the Anther is a outgrowth like structure. Anther is a outgrowth like structure which appears as oval spherical shaped structure. At this stage it is a mass of meristematic cells. At this stage it is a mass of, this is totally mass of meristematic cells which is surrounded by a single hard outer layer is present. That single hard outer layer is also known as epidermis. Next one. Here, firstly, the vascular tissue is developed in the middle regions of the Anther. Where is the vascular tissue is mainly present in the middle regions of the Anther? Here we know that the Anther, in the diathecous Anther, 2 thecas or 2 lobes are present. This is one lobe, this is one lobe. The two lobes are jointed at the point of connective tissue. This is nothing but a connective tissue. The connective tissue is mainly made up of xylem and phloem. You know that xylem and phloems are combined together to form to create the vascular bundles. The connective is mainly made up of xylem and phloem. Through this xylem and phloem, transport of water and transport of the food materials are supplies to development of the pollen grains and microspores. Next one. Here, very important, how to develop the Anther walls in Anther. At the 4 corners of the Anther, this is one corner, this is one corner, this is one corner, this is one corner. At the 4 corners of the Anther, below the epidermis, exactly at the hypodermal position, a group of the cells are developed. These cells are it consisting of dense cytoplasm and large visible nucleus is present. These cells are known as archisporial cells. Here archisporial cells are developed at the 4 corner regions. Later, the archisporial cells are it undergoes periclinal division to form primary parietal cell towards epidermis. Primary parietal cell towards epidermis and primary sporogenous tissue towards center. Next one. The primary parietal cell and primary sporogenous cells are later it undergoes some later it undergoes further divisions and developed into entire structure of the anther except at the epidermis. This is also one more important point. Here, the primary parietal cell and primary sporogenous cells are developed from the archisporial cells. Next, what about the characteristic features of the primary parietal cell? The primary parietal cell located towards the epidermis. The primary parietal cell, it undergoes periclinal divisions to form a series of 3 to 5 layers, making the walls of the anther. Next one. Here, totally how many wall layers are present? 3 to 5 layers, making the walls of the anther. Out of the 5 layers, the first layer present towards the epidermis, which constitute the endothelium. Below the endothelium, 1 to 3 cell layers are also known as middle layers. Below the middle layers, the innermost layer of the anther is also known as tapetum. Later we will discuss the entire characteristic features of the tapetum. Next one. Here, endothelium 1 layer, middle layers are 3 layers, tapetum is 1 layer. Total how many wall layers are developed? 5 wall layers. That 5 wall layers of the anthers are developed from the primary parietal cell. Next one. The primary sporogenous cells, it undergoes mitotic divisions to form sporogenous tissue or sporogenous cells. Here, remember the one important point. Primary sporogenous cells, it undergoes mitotic division to form sporogenous tissue. This is the totally sporogenous tissue present inside of the pollen sac, present inside of the microsporangium. Later, the sporogenous tissue, it undergoes mitotic division to form microspore mother cells. The body of the microspore mother cells 2N, the body of the sporogenous tissue 2N, the body of the primary sporogenous tissue 2N, the body of the primary parietal cell 2N. Next one, the microspore mother cell, it undergoes meiosis division to form pollen grains. The body of the pollen grain N, the body of the pollen grain N. In this way, from this archisporial cell, different wall layers are developed in structure of the anther. Now I will explain the structure of the anther. This is exactly NCERT textbook matter. A typical angioscopic anther or the transverse sections of a typical microsporangium appears as nearly circular outgrowth like structure. The anther is surrounded by 4 wall layers such as epidermis, endothelium, middle layer and tapetum. The anther is surrounded by 4 wall layers such as epidermis, endothelium, middle layer and tapetum. Now we are discussing the each and every layers of the anther. First one is epidermis. It is a outermost layer of the anther. The epidermal cells are arranged compactly without intercellular spaces. It is a one cell thick. The cells present between the two pollen sac water. The cells present between the two microsporangium thin, which constitute the stomium. The stomium is mainly helpful for dehiscence of the anther. The stomium is mainly helpful for dehiscence of the anther. At the time of the anther maturation, the stomium cells are completely degenerate and helpful for dehiscence of the anther. This is about the epidermis characters. Next one, the second layer of the anther is also known as endothecium. Endothecium is present below the epidermis. The endothecium cells are arranged compactly without intercellular spaces. At the anther maturation, various changes takes place in the different walls of the endothecium. Here, at the anther maturation, various changes takes place in different wall layers of the endothecium. For example, this is one endothecium cell. This is the outer side of the cell. This is the inner side of the cell. Here this is nothing but radial walls. The outer side of the endothecium cells are thin. The inner side as well as radial walls of the endothecium cells are thickening due to the presence of fibrous thickenings of cellulose. Once again listen. Here at the anther mature, various changes takes place in different wall layers of the endothecium. The outer side of the endothecium cells are thin. The inner side and the radial walls of the endothecium cells are thickening due to the presence of fibrous thickenings of cellulose. Actually, due to the presence of fibrous thickenings of the cellulose, the endothecium cells act as a hygroscopic nature. What is meant by hygroscopic nature? Hygroscopic means the cells it absorb the water very fastly, and also the cells lost the water also very fastly. This is nothing but hygroscopic characters. Actually here 4 wall layers are present in the anther. Out of the 4 wall layers, which anther wall layers are? It contains hygroscopic nature only endothecium. Why the endothecium cells are act as a hygroscopic nature? Because of due to the presence of fibrous thickenings of the cellulose, the endothecium cells are act as a hygroscopic nature. At the anther maturation, the endothecium cells are mainly helpful for dehiscence of the anther. Here, at the anther maturation, the endothecium cells are mainly helpful for dehiscence of the anther. This is about the endothecium characters. Next one, the third layer is also known as middle layers. Here, 1 to 3 celled middle layers are present below the endothecium. Otherwise, 1 to 3 celled middle layers present between endothecium and tapetum. Here, what is the main important functions of the middle layers? The middle layers are mainly made up of parenchymal tissue. The middle layers are mainly made up of the parenchymal tissue, which store the food materials. At the time of anther maturation, the middle layers are completely consumed. At the time of anther maturation, the middle layers are completely degenerated. That's why the middle layers are act as a ephemeral layers. The middle layers are act as a ephemeral layers. Ephemeral layers. Ephemeral means short-lived. Ephemeral means short-lived. Ephemeral means short-lived. Next one. This is also known as middle layers characters. Next one. The last layer of the anther is also known as a tapetum. The innermost layer of the anther is a tapetum, which encircles the sporogenous tissue. The tapetal cells are encircle the sporogenous tissue. Initially, the tapetal cells it consists of dense cytoplasm and one or two nucleus are present. Initially, the tapetal cells are diploid, but they became, for example, this is nothing but one tapetal cell. Initially, the tapetal cells are diploid, but they became polyploidy. And multinucleated. Endomitosis or a free nuclear division. Very important point. Why the tapetal cells are act as a diploid nature? Why the tapetal cells are act as a diploid nature? In that tapetal cell, karyokinesis is occurred, but not followed by the cytokinesis division. That's why inside of the tapetal cells, more than two nuclei are present. How to more number of the nuclei are developed inside of the tapetal cells? Because of karyokinesis division is occurred, but not followed by the cytokinesis division. This type of the division is also known as endomitosis division or free nuclear division. Next one. What is the main important functions of the tapetum? Tapetum cells store the food material. Tapetal cells are store the food material. The tapetal cells are which observe the food materials from the middle layers. The tapetal cell nourishes the development of the tapetal cell nourishes the development of sporogenous cells, or are microspore mother cells, or are pollen mother cells, or are pollen grains. It is very important point. What is the main important functions of the tapetum? The tapetal cells nourishes the development of the sporogenous tissue, or are microspore mother cell, or are pollen mother cell, or are pollen grain. At the time of the anther maturation, the tapetal cells are completely degenerated. That's why tapetal cells are also act as a ephemeral cells. How many wall layers of the anther act as a ephemeral cells? 2 wall layers. That 2 wall layers are also known as middle layers and tapetal cells. Why the tapetal cell? It consists of 2 nucleated condition because of karyokinesis is occurred but not followed by the cytokinesis division. This type of the division is also known as endomitosis or prenuclear division. Next one. The tapetal cells are also secrete the enzymes and hormones. The tapetal cells are also secrete the enzymes and hormones. Before degenerating of the tapetal cell produces the probisc bodies. Here, probisc bodies. This is also nothing but probisc bodies, which provide the probisc bodies which is surrounded by the sporopollenin. That's why the probisc bodies are also known as ubisc bodies. The probisc bodies are also called as ubisc bodies. At the time of tapetum degenerates, the ubisc bodies are released into pollen sac. The ubisc bodies are released into pollen sac. Next one. What is the main important functions of the ubisc bodies? Ubisc bodies participates in the formations of outer wall layers of the exine of pollen grain. Once again, revision. Ubisc bodies are mainly helpful for formations of outer wall layers of exines of pollen grain. Next one. What is the main important functions of the tapetum? The tapetal cells provide the food material for the development of the sporogenous tissue, microspore mother cell, pollen mother cell, and pollen grain. One more important function. Tapetal cell secrete the sporopollenin and pollen kit materials. Tapetal cell secrete the sporopollenin and pollen kit materials. Next one. Tapetal cell secrete the hormones and enzymes. Tapetal cell secretes the hormones and enzymes, and tapetal cell mainly helpful for formations of ubisc bodies. This is about the tapetum main important function. Here, very important 5 marks question. Now, once again, I will briefly explain the structure of the anther. A typical angiospermic anther, it appears as nearly circular in outline. Each anther surrounded by a 4 wall layers such as epidermis, endothelium, middle layer, and tapetum. First of all, what is meant by epidermis? Epidermis is the outermost layer of the anther. The epidermal cells are arranged compactly without intercellular spaces. The epidermal cells are one cell thick. The epidermal cells are one cell thick. The cells present between the two pollen sac. The cells present between the two pollen sac or two microsporangium. These cells are thin, which constitute the stromium. The stromium is mainly helpful for dehiscence of the anther. At the time of anther maturation, the stromium cells are completely degenerate. Through these stromium cells, the pollen grains are released to outside. Second wall layer is also known as endothelium. Actually here, the endothelium present below the epidermis. The endothelium cells are arranged compactly without intercellular spaces. Next one, at the time of anther maturation, various changes takes place in different wall layers of the endothelium. The outer layer of the endothelium cells are thin. The inner side of the endothelium cells and radial cells are thick due to the depositions of fibrous thickenings of the cellulose. Actually, the endothelium cells act as a hygroscopic nature. Why the endothelium cells are act as a hygroscopic nature? Because of due to the presence of fibrous thicknings of the cellulose. At the time of anther maturation, the endothelium cells are mainly helpful for dehiscence of the anther. This is about that endothelium. Next, middle layer. 1 to 3 celled middle layers are present below the endothelium. Otherwise, 1 to 3 celled middle layers are present in between endothelium and tapetum. The middle layer cells are made up of parenchyma cells. The parenchyma cells are store the food material. At the time of anther maturation,